This is just one of the attempts to try to bring otherwise huge, monumentally large part of what we know from the world of organic chemistry into relatively short video presentations. So before I start, let me just be perfectly clear. This is not really intended to teach you organic chemistry, but rather to give you a little bit of ideas as where the organic chemistry fits into human body and to import it into lectures or video presentations that will come a little bit later. In a section about inorganic chemistry, I mention it very clearly. There is no molecule that fits into organic chemistry without at least one atom of carbon. Of course, it is going to be just a single carbon atom for something that is very simple as a methane gas, carbon binding for hydrogen atoms. All other molecules that you and I will share and talk about are going to be way more complex than just CH4 or methane. So there are two ways of writing down sometimes excessively large and complicated formulas. Either we're going to see it like this with all atoms written out, or perhaps we can see it in a somewhat shorthand standard where there is no need to introduce carbon at these 6 points of hexagonal structure. Neither there is a need to put letter H or symbol for hydrogen, because they are compounds that are commonly encountered in organic chemistry. So we're trying to make our life a little bit easier and much more, how to say it, simple to handle. Apologize for this kind of like just looking for a word is sometimes a little bit harder, but let's move on. In the world of organic chemistry, you and I will have to talk a little bit about three basic types of molecules that we will be encountering within human body. The first group that our discussion will start with will be discussion about carbohydrates. Then we're going to be talking a little bit about lipids, and ultimately we're going to be saying a few words about proteins, and perhaps at the very end of this recording, few words about adenosine triphosphate, which also fits into organic world. So let's start and let's immediately decipher the idea. Why do we call certain group of molecules carbohydrates? As we know, carbo stands for carbon, hydros is water. So basically hydrated molecules containing carbon. They are our best known sugars. So what kind of sugars we have inside a human body or in natural form? We're talking about monosaccharides, so single molecules having all the characteristics of carbohydrates. And within this group, we're either talking about molecules containing 5 carbon atoms, therefore we call them pentose sugars, or we're talking about monosaccharides having 6 carbon atoms within a molecule, and they're called hexose sugars. Hex refers to 6 carbon atoms. Let's immediately sort out those 2 pentose sugars having 5 carbon atoms within a molecule. They're typically found within the molecules of DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So these 2 sugars are deoxyribose and ribose, depending whether it's observed in a molecule of DNA or RNA. So although the DNA and RNA contains sugar, they're of no nutritional value for us, because simple thing is human body does not possess set of enzymes that would eventually break down the pentose sugar and convert it to some energy carbon dioxide, and water. Therefore, pentose sugars are practically quite stable and quite safe within human body. On the other hand, it's a group of hexose sugars that we use more regularly. And of course, there are three of them. There are galactose, fructose, and glucose. So we have here glucose and fructose examples on the left side of the screen, and you can see that their formula is CHH12OH for both molecule of glucose and fructose, which could confuse us that practically we start seeing them as exactly the same molecule. When we add galactose, it is going to be the same CH612OH molecule, and we're going to start thinking about where is the difference between the three of them. The difference is practically very simple. Arrangement of different attachments to carbon atoms is not the same. So we see here a little bit of a difference if we go with standard shorthand writing of these two molecules, that glucose and fructose really don't look the same, although their short formula appears to be the same. So they're almost like isomers of each other. And these are sugars, so monosugars, galactose, glucose, and fructose, that the body simply can't wait to get into it because we have a set of enzymes capable of breaking them down and converting them to energy. Well, do they come in a natural form on their own? The answer is practically no, because on this example, on this slide, we can actually see what the table sugar or sucrose is. It has been generated on the basis of chemical reaction between glucose and fructose. Two monosaccharides have been subject to chemical reaction which was called a dehydration synthesis. So you can see that the fructose donated hydrogen, while the glucose donated hydroxy group, which combines to form a molecule of water on the far right of the screen. And as a result of dehydration of these two molecules, additional bond was generated through this oxygen atom, and these two molecules of monosaccharides now combine into a molecule of sucrose or table sugar, which, as you can see, is not just a pure sum of these atoms, C12, 12 carbon atoms remain, but H24 actually would become H22, and 12 oxygen atoms is now down to 11. That is because we have subtracted the molecule of water through dehydration synthesis. Now, what's going to happen when we take a spoon of sugar and dissolve it in water? We're going to subject the table sugar to hydrolysis. So with addition of molecule of water, the bond between glucose and fructose will be broken, and two monosaccharides would be released, with the body having a perfect set of enzymes to dissolve it and to further metabolize it and turn it into energy that we need, plus having a two byproduct, carbon dioxide and water, at the complete breakdown of molecule of glucose or fructose or galactose. So essentially, we're to see that sugars, saccharides, are coming not just in the form of mono or disaccharides, as the sucrose table sugar is. We're also seeing polysaccharides, where the same monomer of glucose is part of the molecule, which appears to have no ends, including those multiple sideways related branches. Basically, in a natural form, that is starch, and we know that carrots or potato would be rich rice as well with starch, but also plants would have this polymer. However, we call it cellulose. So where is the difference in the polysaccharides? Well, the difference is that for starches, we are having enzymes capable of separating and breaking down individually each of these hexagonal structures, molecules of glucose, separating them from the chain and individually digesting them down. For cellulose, aka fibers, we do not have enzymes, so the cellulose, even if ingested, practically becomes excreted from the body in a very same form as it has been ingested. Sugars, of course, are going to be considered the very important fuel, food that we can quickly intake, very quickly metabolize it, and very quickly generate the energy. As we see on a number of different sports where competitors during the break, during the short timeout, would opt for some kind of source of energy. And sometimes it's banana, sometimes it's a bite of some kind of bar, but generally that is intake of sugar, which quickly converts itself into energy. A little bit more steady as building blocks of the body are lipids. And a group of lipids is also quite complex. So I'm repeating again that we do not want to make sure that, that we don't want to make this kind of review of the organic chemistry, but rather some kind of introduction to basic principles of organic chemistry. Lipids are present in the human body on average in an adult lean person between 18 to 25%. One will say, wow, it's a significant percentage. Of course it is, but we do not necessarily think about lipids being part of our adipose subcutaneous tissue, but rather we have to take a closer look to individual cell and its membrane, which is built practically almost exclusively on the basis of lipid molecules. So a variety of lipids in the human body is also quite staggering, and we would not have time, neither are we going to have needs to address all of them. Essentially, when we talk about lipids, we're going to say that one of the most significant groups of molecules that we're finding within a human body are so-called triglycerides. What is hidden in the name of triglyceride? We can see it here already on this diagram. There is a molecule of glycerol, which is organic alcohol, having these three hydroxy groups. And each and every one of them would be able to interact with a fatty acid molecule. So when they interact, it is going to be the same dehydration synthesis between the glycerol and the fatty acid. So in this case, it is palmitic acid molecule with the glycerol, getting us to a level of monoglyceride. When the second hydroxy group would do similar type of dehydration synthesis reaction, it'll become diglycerol. And when the third one is also subject to dehydration synthesis, and the third fatty acid is added, as we see on the diagram below, palmitic, stearic, and oleic acid, basically now we have a full molecule of triglyceride. So three fatty acids being attached to a single molecule of organic alcohol called the glycerol. If you take a look, you're going to see that we have most of these fatty acid molecules utilizing a single covalent bond between carbon and hydrogen atoms. Only on the oleic acid, we can see here that between two consecutive carbons, we have formed a double covalent bond, obviously not having a hydrogen here or here because all pairs of electrons have been already shared. So what's going on here? We're talking about principle of some fatty acids being called saturated. And saturated usually means that they are seen in an animal's origin, humans included. Of course, that's our adipose tissue or fat. And we're talking about all bonds being single covalent bonds that we're seeing within a molecule. If we're talking about appearance of a single covalent bond, then we're talking about mono unsaturated fats. They would contain fatty acids with at least one double covalent bond between two fatty acids and carbon atoms. They are not completely saturated with hydrogen atoms. For that reason, we're calling them monounsaturated. Where do we find them? Olive oil, peanut oil are rich in triglycerides that are having monounsaturated fatty acids. If we have more than one double covalent bond between fatty acid and carbon atoms, basically we're talking about polyunsaturated fats. They could be also seen in a natural form. Canola oil contains them, corn oil, sunflower oil, soybean oil are all containing polyunsaturated fats. So essentially, we know that fats are excellent energy storage, and we can break them down, especially in the cases of prolonged lack of food or starvation. Basically, we're going to rely on our own internal reserves. They're not so quickly metabolized into energy. It's going to take a little time until we start burning our own adipose tissue, but it is definitely possible. So why do we have need to understand triglycerides if we're talking about them as source of energy and something that's more pertinently present in the biochemistry? The answer is this. With a minor modification where the third fatty acid is replaced with phosphate group, basically we're talking about completely new form of lipid molecule, and we're calling it different name. We're calling it phospholipid. Everybody knows that oil and water do not mix. Basically, oil is hydrophobic. It repels molecules of water and doesn't allow them to interact. Therefore, that's why the lipids are quite stable when interacting with water. But with the substitution of a single fatty acid with a phosphate group, basically we have made this molecule gaining its so-called polar head, which essentially is where the phosphate is added to glycerol. The polar head is hydrophilic, which means this molecule is easily interacting with water molecules around its polar head, while its fatty acids that are making nonpolar tails are going to be repelling water molecules. So one can think about, okay, what's the usefulness of this information? Well, the phospholipid is going to be essential building block that we have to utilize in order to build cellular membranes. Our cells need to have some kind of semi-solid shield that we call the cellular membrane, which is made of two layers of phospholipid molecules that are arranged so that polar heads are facing out of the cell. I'll just put letter O, and opposite layer is having phosphate heads that are directed into the cell inwards, which means water that comes in contact with the cellular surface is not repelled. Water that fills the inside of the cell is also not repelled away from a membrane. But with these two layers of fatty acid tails that are nonpolar, water would not be so welcomed in a core part of the cellular membrane. You will see later on, it doesn't mean there is absolutely not a single molecule of water, but water would be able to pass the cellular membrane going both direction out and in, but it will not be retained within the cellular membrane. And this is that kind of idea of non-polar tails being hydrophobic. So they would not completely block the movement of water molecules across the cellular membrane, but they would, to some extent of this word, control it. Of course, triglycerides and phospholipids are not only two classes of lipid molecules. Here we can have another example of molecules that are called steroids. What is common quality and common denominator for them? You can see that all of them have those carbon rings, whether they're made in the form of hexagonal rings or pentagonal rings. They're connected to each other. So practically, if you just glance through this diagram, you're going to see that these molecules look very comparable to each other. Again, what makes them being different is that attachment of different structures at the different points of these kind of ring molecules are going to make it totally different. So here's an example of what the cholesterol would look like. And we know that cholesterol is not necessarily our greatest ally when it comes to health and measuring the total amount of cholesterol that circulates within our body. However, its related molecule is something that's quite the opposite. It's estradiol. It's estrogen or a female reproductive or sex chromosome. Also, very similarly looking with some minor modification is the testosterone. Again, reproductive, but this time male reproductive hormone. Or finally, when we have example D, which illustrates the same type of carbon ring, we're now seeing it as a cortisol, basically a hormone produced by adrenal gland. Obviously, minor modifications are going to be of certain interest, more so for pharmacology, for pharmacodynamics, but we want at least to understand where these molecules would fit, into which category they would fit. Certainly, it would be too quick for us to move to proteins before saying that other form of lipid molecules do exist. And we're seeing some of the vitamins, if they are lipid-soluble vitamins, like vitamins A, D, E, and K, they're going to fit also into this group, which shows huge variety of lipid molecules. But another important building block of a human body, being present in a range 12 to 18% in an average adult, are, of course, proteins. And this is where we end up having the widest possible range of varieties. Firstly, let's find out what protein molecules are made of. We know that they must be made of some kind of smaller individual building blocks, and they're called amino acids. So on this diagram, you can see what is the group which is added to a carbon atom, which we call amino group, a single nitrogen with two extra hydrogen atoms. That is amino group. So if we're to write it down quickly, we're going to say it is NH2 group, and all amino acids certainly must have it. Otherwise, they would not fit into this classification. On the other side of the molecule, we have group COOH, which is essentially like group that is something that we see as the carboxyl group for typically found in organic acids. Again, in short form, we're going to write it like this, COOH, without need to necessarily think about where exactly are covalent bonds made, which one is double, which one is single, but that is the carboxyl group. So amino group NH2, carboxyl group COOH, and of course, then the rest of the amino acid molecule would look different, and that is symbolically illustrated here with box saying letter R. That's the radical form, which could be made of a variety of different carbon atoms just being linked to a chain. Therefore, amino acids come from sources that we consume on a daily basis, and some of them we are able to synthesize within our own body. Those ones that the human body can synthesize are called the non-essential amino acids because we can make them whenever we need them. But those ones that the body is unable to do, we have to have their intake into the body, and they're considered to be essential amino acids. So out of 20 amino acids, 10 are essential, 10 are non-essential. And keeping them at the level of amino acids essentially is enabling us to start playing with them, so to say, just like using them as individual Lego blocks to start building structures that would be looking progressively bigger, larger, and will get a variety of different functions. Just to name a few, for example, proteins that are seen within our muscles are actually called the contractile proteins. Without them, muscles would be unable to do contraction. Fast forward to the other side, there are some proteins without which we would be suffering very severe consequences, even of the most mild form of common cold. Of course, now I'm talking about some proteins that are having a different functional role. They become antibodies, and they keep our immune system up to speed, up to certain levels, enabling us to, from here and there, get occasional sickness, but to come out of it in a matter of 2-3 days' time. So how is this possible that amino acids are building progressively longer chain of amino acids until they become functional proteins? It is the same thing as what we have said for monosaccharides generating disaccharides and reversal disaccharides being hydrolytically changed to a level of 2 monosaccharides. So we're using the dehydration synthesis, removing between carboxyl and amino group molecule of water. We're creating newly formed bond, which we call now the peptide bond. And instead of 2 amino acids, we're having a small protein that could be called a dipeptide because it's made of 2 amino acids only. And of course, we keep going with same reaction of dehydration synthesis, and we can progress from 2 to 3 to 5 to 10 to hundreds of amino acids that are linked into chain. And this is where we can say the process of building proteins have been completed. That statement would be quite false to make. Yes, we have generated somewhat longer and somewhat more complex chain of amino acids, but we have only achieved to reach the level of primary structure of protein. Amino acids of a different kind are properly lined up with a proper sequence, and they are not dipeptide or tripeptide. Now they're polypeptide. Poly refers to practically countless number of amino acids thrown into a chain. If they would stay at this level, proteins and their function will be quite predictable, but they would undergo secondary structure, whereas you can see here that chain of amino acid is going to be twisted, folded, and creating now some form of helix, which obviously looks very different compared to what we have seen in the form of just simple plain sheet. We can think about secondary structure, either creating a helix like a coil, or to think of more complex form of protein, basically being like a pleated sheet. where we keep folding it, and we're inevitably now changing some kind of dynamics of the molecule. Even that is not the end of the story. We can go into a tertiary shape where the protein essentially gains the three-dimensional shape of polypeptide chain. So either alpha, helix, or pleated sheet, we're still two-dimensional. With many more twists being added, practically we're now seeing a molecule of some more complex protein being like a little body, being three-dimensional, having its height, its width, its depth. And even if we would stop here at this level of tertiary level of organization of proteins, we would realize very quickly that this adds to a variety of proteins and their different functionality. But we need to complete their final step, quaternary structure, which requires sometimes two, sometimes four, sometimes more of different proteins, regardless of which level of organization they have achieved, to kind of mix up together. And this clearly identifies two different chains, one colored blue, one colored brown grayish, obviously two different proteins with tertiary structure being now forced to come together, essentially is what makes it quaternary structure, and that even adds more to a variety of proteins. For example, quaternary structure, good example would be a protein that is composed of two short chains and two long chains. Of course, these long lines represent long chains, shorter lines represent shorter chains. So we can see that I drew here 4 different lines. And that is something that it would be typically a structure of our antibodies, proteins, way complex with their structure, but also unbelievably reliable when it comes to their function as part of our immune system. To talk about organic chemistry and to avoid any discussion about enzymes would be a bit of a problem, not to say sin, but we know that enzymes are also extremely important compounds that we're finding within our body. What is their typical best quality? Enzymes actually facilitate chemical reactions without changing themselves before, during, or after the chemical reaction they were part of. So each enzyme will have its own active site, and it will bind on its active site a substrate. In this case, it is going to be a sucrose, which is disaccharide, table sugar, and will bind a molecule of water. This enzyme is called a sucrase. Please note that OSE ending denotes sugars. ASE ending is denoting enzymes. That is one tiny rule that we can throw in here, just to reveal part of secret about their naming. Enzyme actively facilitates this chemical reaction, and it breaks down molecule of sucrose into a molecule of glucose and fructose, after which these two individual molecules are released from the active site of an enzyme, and enzyme remains ready for yet another cycle to keep repeating the same chemical reaction over and over again for as long as the substrate, in this case sucrose, is available, or of water. That explains the longevity of enzymes, that we really need them in a minute amount, but we need them. So just as the example for people who are, for example, lactose intolerant, what do we have? It's not natural animosity towards lactose, milk sugar. It is lack of lactase, the enzyme, which doesn't allow us to break the lactose into monosaccharides. As a result of that, disaccharide keeps passing within our digestive tube, within our gut, without having a chance to be chemically broken down, and as such is a subject to different processes of its biochemical degradation, resulting in attracting a little bit of additional water, and of course creating a bit of gas and creating a number of different digestive difficulties, including the diarrhea, which would be one of the common problems that people who are lactose intolerant are experiencing. So enzyme is stable and steady, gets in and out of chemical reaction, being practically unchanged, and makes itself available and ready for another round of same chemical reactions. The complexity of organic chemistry perhaps cannot be better illustrated anywhere else than just taking a look at the single molecule of deoxyribonucleic acid, in short, DNA. When we say that this molecule is practically endless, we immediately develop some kind of fear of how are we ever going to understand what it is. Basically, we have to break it down. We have to do some kind of piecemeal format in order to understand that it's just excessively long molecule, but being built of very predictable 4 types of base building blocks. So let's see these 4 building blocks, what they're called. They're named thymine, adenine, cytosine, and guanine. Obviously, with this hexagonal carbon ring, they are fitting nicely into organic chemistry. To either one of them, we're going to have in a molecule of DNA, deoxyribose sugar. It's here. It's the same what's here. It's the same what's here. It's the same what's here. And then we have each of these nucleotides. Nucleotide is composed of nitrogenous base, thymine, adenine, cytosine, or guanine, linked to a single molecule of deoxyribose sugar, which in turn is linked to another phosphate group, same here and here, as well as here at the end next to guanine. So the term nucleotide is essentially one nitrogenous base. If you see here, each thymine, adenine, guanine, or cytosine will contain some nitrogen as part of its molecule. Hence, it's easy for us to call them nitrogenous bases. Linked to pentose sugar, deoxyribose, which we don't break down as we don't have enzymes capable of doing it. And of course, the phosphate group, which is added to individual nucleotide. So if I would draw 2 lines like this across the screen, essentially I could present them as 4 individual nucleotides, one after another. But the reason Illustrator opted for some extra dotted lines between thymine and adenine and cytosine and guanine is to show that they establish those interesting bonds that are hydrogen bonds, like what we discussed when talking about molecular water. So not exceptionally strong bond, but still good enough to keep thymine linked to adenine or adenine linked to thymine. That's the only possible combination. Thymine cannot link to guanine, neither cytosine, only to adenine. And also cytosine and guanine are only possible combination between 2 nucleotides. Since the DNA, deoxyribonucleic acid, is composed of 2 parallel running helices, chains, you can see here that on this simplified illustration between 2 different strands of the DNA, you only have this possible combination: cytosine bonded to other strand of a DNA through guanine containing nucleotide with a hydrogen bond, thymine to adenine, cytosine to guanine, adenine to thymine, cytosine to guanine, thymine to adenine, cytosine to guanine. So we can only see this: adenine loves thymine, while cytosine and guanine are liking each other, and they were able to establish the bond. So in short, This is part of the DNA. We just try to understand its basic structure, individual nucleotide containing a nitrogenous base, phosphate group, and pentose sugar. If we're talking about ribonucleic acid, RNA, it would look very much the same, except the sugar will not be deoxyribose, it would be just a ribose, hence the ribonucleic acid. And also, we're not going to have thymine. Instead of thymine, molecule of RNA will have another nitrogenous base, which is called the uracil. So thymine would not exist in RNA, and it would be replaced by uracil. Finally, as I mentioned in the introductory part, we have one interesting organic compound to address. It is adenosine triphosphate. Adenosine, okay, sounds already familiar. Adenine. Let's take a look at previous slide. Adenine being part of nucleotide of a DNA, and the same nitrogenous base is found within a molecule of ATP. That's fine. Okay, so what's next? Sugar, we can say this is ribose. Okay, it's not deoxyribose, so we're not seeing it the same as on the previous slide. It's the ribose. And look at this. Instead of one, we have 1, 2, 3 parts of phosphorus containing molecule. Hence the name adenosine triphosphate. And on this diagram, you also see the squiggly line between oxygen to phosphate at 2 different locations. Basically, it's a bond which is easily broken. And when this bond is broken, it releases the energy. So for example, if this bond becomes broken, this phosphate group is released, and adenosine triphosphate becomes adenosine diphosphate, ADP, having just 2 phosphate groups. And perhaps if we continue with need for energy, another phosphate bond will be broken, so another phosphate group will be released, and adenosine triphosphate or adenosine diphosphate will now be scaled down to adenosine monophosphate, AMP. And if we would put a period sign here saying what's going to happen to AMP, basically it's now having no energy, we're going to reverse the reaction. Basically, there will be moments in time when the body doesn't need to have a massive energy expenditure, but rather we are rebuilding our energy reserves. The phosphate group being released doesn't mean it's going to be lost. It is going to be re-imported and rebonded. So a little bit of energy, of course, will be used to bond second, and more energy will be required to bond third phosphate group. So we're to kind of recycle adenosine triphosphate by converting it all the way down to adenosine monophosphate, and with a little bit of energy investment, we can rebuild adenosine triphosphate, which really becomes something that's very important to know and to understand. It is considered to be that kind of stored energy, something like a battery, something like, fuel, which we have not ignited, which we have not yet burned. We're waiting until the time is for this kind of extra energy requirement to start using it from the depot. Once again, I believe that we are seeing this as just a review of some principles of organic chemistry, not to be considered or taken as some kind of organic chemistry lecture, but rather just a review of important and interesting items that we have to keep using for the rest of our studies. If you recall the introductory part of physiology, we have gone through atom, molecular level of arrangement, both cellular, both inorganic and organic. So the next step that you and I will have to take together would be to address the cellular level of arrangement. We're not going to spend too much time on definition and action and roles of certain organelles within a cell. We're going to talk a little bit about them, but in order to apply life to a cell, we're going to spend most of the time focusing on events that are happening on the cellular membrane and the transport that would be allowed so that we start seeing the cell as part of a living organism, living creature. And just to be able to say at the level of single cell, it looks like that. Anyway, thank you for watching this video, and I hope that we're going to spend some good time together in the near future. Thank you.